Great. Thank you so much, Justin, for that introduction. And thank you all for organizing this. It's really helpful and insightful. So I want to thank everyone for coming to my talk today about leveraging mass spectrometry proteomics towards biomedical applications. My name is Aleksandra Petelski, and I'm an assistant professor in the Department of Materials Science and Engineering, and I just started my lab this fall. Just to give a little bit of background about myself, I completed my bachelor's and master's degrees at the Stevens Institute of Technology in 2016. And while I was there, I worked at Stryker Orthopedics, where I learned a lot about additive manufacturing and different material characterization techniques. I also work at the Rockefeller University on more biological-based experiments on inner ear hair cell biology. I then jumped to do my PhD at Northeastern University in bioengineering, where I took up a lot of different positions, and I also learned a lot about mass spectrometry and how to utilize it for the study of proteomics. And now my most recent position, as Justin mentioned, I was a co-founder of a new type of research organization that follows a nonprofit model called Parallel Square Technology Institute, or PTI. Our mission there is to really drastically increase the throughput of mass spectrometry proteomics so that we can enable really large studies that are population level that are currently almost impossible to do today. And so what does my lab do, and what do I want my lab to be doing at UCF? Well, I want to combine my interest in materials engineering and biomaterials and in mass spectrometry proteomics and leverage the two in order to unveil proteomic secrets that can help build better biomaterials and therapeutics. And so what's our motivation here in terms of interpreting biology using mass spectrometry? Well, mass spectrometry is a very valuable and powerful tool that has enabled the study of different types of organs within the human body, as you see here. And as we know, there's lots of different organs, have lots of very disparate functions, such as the human lungs. But within the human lungs itself, there's actually more than 10 billion single cells in human lungs. And you may think they're all homogeneous and performing the same functions, but really in reality, we have a level of the different types of heterogeneity at just the single cells. There's lots of different types of cells doing lots of different functions to help us breathe in just the lungs. And then if you look at just a single cell, the complexity is further induced, whereby we're seeing lots of different proteins and more than 20,000 different proteins, which are the ultimate products of gene expression, performing different functions so that cell can actually do what it's supposed to do within our different tissues and organs. And this collection of proteins is known as the proteome, and it really determines the final cell status and function. And this brings us really to the second point of, well, in some experiments, maybe you want to know about one or two proteins, but in some biological contexts, in order to really gain what the cell is doing at that particular point in time, you really need more of a global proteome measurement where you're measuring thousands of proteins. And that's where mass spectrometry comes in, because it enables us to do that. And both of these things, the heterogeneity at the single cell resolution and the requirements of global proteome measurements, leads us to the fact that we want really at the core of biology, we want to understand how do cells function. And within biomaterials, we have all these different parameters like nanoparticle size, porosity, and stuff like this that you can tune. But how does that affect cell function, and vice versa? How does cell function affect the materials? And here's where I want to combine the use of mass spectrometry to study the cell function in order to influence material properties. So at Northeastern University, I was part of the team that was developing the second platform technology of single-cell proteomics at the Slavlov lab, where we were able to go from single mammalian cells, all the way to get proteomics data. We were able to analyze thousands of mammalian cells in cancer and immune cells that we were able to gain information about thousands of different types of proteins. How did we really go about to do this? Well, our platform, which we call Scope 2, requires lots of series of different steps, which I'm not going to get into too much, but really in essence, you have to isolate your single cells. There's lots of different sample preparation steps here. And then that undergoes liquid chromatography, tandem mass spectrometry, and where we can acquire the proteomics data and then further data analysis downstream so we can actually understand which proteins are upregulated in different contexts. And this work has been published in several different venues, including we've published recently a protocol of step-by-step on how to do Scope 2 to make it more accessible to different labs. And so at UCF, I want to be able to connect this proteomics insights at the single cell level and also at the bulk level to biomaterial design. And here are really the three different areas I'd like to focus on in my lab. The first area is regaining proteomic homeostasis and dysregulated biological states. In this graph here, I'm showing the protein quality control network, which is really at a bird's eye view of the series of 3 steps or 3 different procedures that are occurring in cells, which are protein synthesis and protein degradation are interplaying with each other. But then also it's really important to understand how do proteins fold and maintain their conformation in order to perform the functions. And this sort of quality control network breaks down in different types of cases, such as musculoskeletal disease and in cancer. And so understanding the proteomics behind of why this breaks down can help us answer the questions of how we can basically regulate back to its former function of being cancer-free or basically having bones being regenerated. And the second area, I want to really understand the relationships between biomaterials and cell function. So when you're developing biomaterials, there's lots of different parameters that you can tune, and understanding that more systematically of let's say we tune a little bit more and a little bit more, how do the cells function and how do different types of cells react to this type of parameter optimization, so that we can progress in tissue engineering and engineer different types of tissue architectures and different types of materials to help bone regeneration, for example, other different types of biological contexts. In area 3, finally, I want to investigate mechanisms behind tissue regeneration. Can we use the cells that are already there in the injured tissue or in the broken bone in order to recruit them in the way so that they can actually start repairing sooner and actually start having repairing of bone or different tissues that's already existing within the tissue itself? And so these 3 different areas are very interdisciplinary. There's lots of different skill sets involved here. And I'd love to talk to people more about this if you're open to it. But for now, I'm going to focus on area 1 for today, in musculoskeletal disease and in cancer, and give you a little bit of a bird's eye view of what I'm interested in. So in musculoskeletal disease, the proteostasis or the homeostasis of proteomics really becomes dysregulated. If we go back to how healthy bone looks like, which we're seeing here, is that there's a delicate interplay between bone formation and bone resorption, where basically the balance is of bones being formed versus bones being broken down. And when they're broken down, they release minerals such as calcium that can be entered into our bloodstream. That's good for us. In musculoskeletal disease, such as an osteoporosis, this balance tips in a direction that we don't want it to tip, where bones are actually becoming bone resorption is being done more so than bone formation, which leads to occurrence of bone loss, and eventually you are losing more and more bone rather than it being formed. This sort of state of osteoporosis is currently not curable, and it also leads to higher risk of fractures. And if you look at different types of bone fractures, we were worried about regaining the bone back, right? But really what happens before trying to regain bone, there's actually immune cells that come to the site and are recruited to do many different functions there. And that's really where a single cell analysis can shine in understanding how bone healing actually can occur within how these immune cells are recruited, what are they doing, and in terms of the bone cells and how do they interact with the immune cells themselves. And furthermore, what's really also interesting and really drives proteomics home and why we would want to study this is, for example, in a hip fracture, scientists have found that in different types of bone, the protein synthesis rates are actually different. And trying to track which proteins are being, activated here is also kind of a challenge that I would like to help solve. And so really the key crux here is that we don't really understand what drives proper bone healing that could possibly help, heal fractures or heal musculoskeletal disease. And because they're insufficiently understood, perhaps proteomics by mass spectrometry can help us understand those mechanisms better. And this proteostasis is also dysregulated in cancer. So there's a different type of scaling here that I wanted to show in a normal cell versus a cancer cell. So in a normal cell, we have this sort of mechanisms of a normal protein load where there's a lot of different production of different types of proteins, but they're produced at a rate at where the proteasome or this protein complex that degrades proteins can actually, degrade the proteins that are tagged for degradation at a normal rate. And this helps with homeostasis. In a cancer cell, this is basically misbalanced. Those mutations are causing proteins to be overproduced in cancer cells at a very alarming rate, which overwhelms the proteasome and other degradation machinery, and therefore the cancer spreads from a bird's eye view. And this is really different. You can see, and this is sort of a bird's eye view of different protein levels of tumors and tumor tissue, or the tissue that's adjacent to the tumor, or the distant tissue to the tumor. You can see the different protein levels are very different among these different types of tissues, whereby there's a lot more proteins that are in green here, meaning they're really more upregulated in these tumor cells. But where does single cell analysis really come in here? Well, if we talk about how cancer starts, it really does start from a founder cell, which expands and actually it keeps dividing, and it diversifies into different types of cells here. And once a patient is treated with different types of drugs and chemotherapy, most of the cancer cells are killed, and at a cancer screening, they are declared cancer-free. However, sometimes cancer cells, a specific type that we don't understand, do stay behind and are because they're resistant to the drug treatment, and they're actually later on expand and diversify again, causing cancer to relapse into the patient. Why does this happen? Why is this happening? We don't really understand. And so single cell analysis can help us discern the proteomes of these different types of cancer cells, and targeting this degradation machinery can perhaps be key towards solving drug resistance. Okay, so this is where I want to talk about a little bit about building collaborations at UCF. So I'm in the Materials Science and Engineering department, as I've mentioned. I've reached out to the UCF Burnett School of Biomedical Sciences and the College of Medicine and AMPAC. But really in the future, I want to be focusing on the three areas, as I mentioned, focusing on tissue regeneration and harnessing existing tissues to build biomaterials and studying stem cell differentiation. But I also want to dig into more of computational analyses of deep proteomic data sets in the future, and perhaps improve the technology behind delivery of peptides or proteins into a mass spectrometer. And I really want to bring this emerging technology of mass spectrometry to UCF is my ultimate goal. So thank you so much for giving me this opportunity to talk a little bit about my research interests. Thank you to the Dean, thank you to the College of Engineering and Computer Science, thank you to the Department of Material Science and Engineering, and thank you to the Solvov Lab at Northeastern University. So if you have any questions, I'm happy to take them now, or if you want to have a longer chat, here's my email, and you can also find me in my office. Thank you so much.